In the previous session, I discussed about the various aspects of prokaryotic translation, including pre-initiation, initiation, elongation, and termination. In this session, I'll be discussing about the translation of eukaryotes and eukaryotes. Particularly in this session, I'll be talking about the initiation aspects. The objective of this session is to describe 4 steps of initiation of translation in eukaryotes. Okay, so like in prokaryotes, it is almost similar to prokaryotes, except so there are many factors are there, initiation factor, weather initiation factor, and other factors. Okay, of course, there are some minor differences are there. So translation, when you talk about the translation, like as usual, like prokaryotes, so constitutes of initiation, okay, elongation, okay, then termination, okay, and post translational modifications, okay. So these are the various aspects which takes place in the translation aspect. Okay, then as far as the translation is concerned, there are the various steps. These are the ones which are various steps in various stages which is taking place. As I said, in this session, I'll be taking up the initiation aspect. Okay, then here the first one is, as I said, it is initiation. In the initiation, so what are the steps which are taking place? So, as far as initiation are concerned, there are 4 steps are present. So in the initiation, 4 steps we can say, which can be divided into 4 steps. The first step of the initiation is, so binding of initiator, okay, transfer RNA to 40S ribosome. Okay. The second one is preparation of okay, messenger RNA. That is the second step. Third one is scanning taking place. Scanning of messenger RNA by, okay, 40S. Then finally the formation of so 40S complex, formation of 50 ATS complex. Formation of ATS complex. So this is the first one, binding of transfer RNA. Second one is preparation of messenger RNA. Third one is scanning of messenger RNA by 40S. And finally, so there is a formation of ATS complex. Okay. Then what happens here when you starting the initiation? Let us presume we are starting the second round of the translation. Okay. So that is what we say is a fresh round of initiation. So we are starting the fresh round of the initiation. So what are the things which takes place? So again, if you have to start the initiation, so that can be categorized into A and B. So here what happens? In the here ribosomal dissociation takes place. Ribosomal dissociation, okay, ribosomal dissociation. Then dissociation to what? So there are 2 units are there, 40S and 60S. Here the second one is release of messenger RNA. So these are the 2 aspects so which take place. So fresh round of initiation. Okay. Then what happens? So what are the next steps which takes place in both the aspects, that is ribosome and messenger RNA? Initially 40S is used. The 40S is used, that is converted to 43S pre-initiation complex. So it is 43S pre-initiation complex. Okay. So that is PIC. We call it PIC. It is PIC. Okay. Then simultaneously the messenger RNA. So various factors are added. Okay, added. So association with the factors taking place. Then finally, what happens here? In this one, let us see, after adding these factors, so this will have the combination with the 43S. Then leads to 48S pre-initiation complex. The 48S pre-initiation complex. This is what is happening. Okay, then, so we'll take up the 60S later. Okay, as I said, the ATS complex, when you see the ATS complex, so this is messenger RNA. Okay, this is 3 prime. Okay, so this is the ATS complex. So this is 60S and 40S. One thing it must be very clear that the initiation never starts in the ATS complex as we said. Okay, in the prokaryotes, it never starts with the 60S complex. So this is the ATS complex. Okay, then so in the prokaryotes, it is 70S complex. It never starts with the 70S complex. Okay, sorry. Then, so what happens here? So in this next step, the split, as I said, splitting of 40S and 60S takes place. So here, there is a binding of the factor in the presence of the factors. What are those factors? The factors we can say is, this is the one factor, factor 3, and there is another factor, so that is, say, is factor 1A. These factors we call it, so eukaryotic initiation factors. These are eukaryotic initiation factors. Okay, eukaryotic initiation factors. Okay, so this is the EAF3 and EAF1A. So I will not be again and again mentioning that eukaryotic initiation factor. Simply I will be mentioning about the number of the particular factor so that it will be easier to us. Okay. Then the association of these factors is taking place. What are these factors which are associated in their presence? ATS is converted to 40S and 60S. So that is 3 and 1A. So they combine with the 40S complex. So this is what is happening in the first step. This we can say this is happening in the first step or starting of the first step. Okay. So leading to the, what will happen is leading to the here, the formation of the 40S. Okay. Then there is this is the factor 3. The factor 3 is as large as 40S. Okay. Then also the binding of 1A is taking place. Okay. So this one is 40S. This is 3. This is 1A. Let us take up the first step. In this first step, as I said, the binding of transfer RNA to the 40S is taking place. Here what is happening in the first step? Binding of initiator transfer RNA, okay, so to 40S complex. So that is the first step which is taking place. Okay, so before that, so there are binding of other factors which will take place. What are those factors? The factors which bind are the 5, EAF5. Okay, then the next one is EAF1. Here it is EAF1. So the binding of these factors is taking place. So you can see here the binding of these factors is taking place. Okay, so again, so I'll draw the diagram. In the diagram, let us see again. We have here it is 40S and so this is the 3 factor 3. It is as large as 40S and it will have association with all the factors. And so then, as I said, there is 1A and 1A binding has taken place. Also, so there is here you can say, here it is 1, okay, then here it is 5. Here it is 5, factor 5. Okay, so these are the various factors which are bound to it. So here in this 40s, this is E site. Okay, so then it is P site and A site. Okay, so one aspect we have to take note of this aspect is this 1A, the binding. So where does it binds? So you can see it binds at A site. It binds at A site. So why does binding at A site? By binding it, it prevents binding of incoming transfer RNA to A site. So that is the purpose. So as I said, the post transfer RNA doesn't bind to the A site, it binds to the P site as was explained in the prokaryotic system. So after that, every transfer RNA has to come to the A site. So 1A doesn't allow binding of incoming transfer RNA at the A site. Okay, then next there are binding of the transfer RNA is taking place. Binding of transfer RNA is taking place. So the binding of transfer RNA, how this transfer RNA so looks like. So this is the transfer RNA. This is the transfer RNA. So to this transfer RNA, so there is a methionine. Here the difference from the prokaryotes. In the prokaryotes it is formylmethionine. In the eukaryotes it is methionine. Eukaryotes it is methionine, and there is also a binding of factor 2 and it will have the GTP. It will have GTP, the factor 2 and GTP. So this factor 2, EIF2, so it helps in the recruitment of, so EIF, this is EIF2, so okay, helps in recruitment of transfer RNA. EIF2 helps in the recruitment of transfer RNA, EIF2 GTP. Okay. So next, this one we call it ternary complex. Ternary complex. This is ternary complex. Okay. In this ternary complex, what we have? The transfer RNA is there with the methionine and EIF2 and GTP. So it binds it. So once it binds it, so let us see what is happening. Again, 40s, then there is factor 3, okay, then there is a presence of 1A, okay, then so there is a presence of 1, okay, and so there is presence of, okay, here, see the same which are bound to it. So this is what, this is 5. This is the 5. Now, let us see what it is. Here it is methionine, and then, so the binding of 2 with the GTP. Binding of the factor 2 with the GTP. So, the another difference from the prokaryotic system is the transfer RNA doesn't bind immediately. After some time binds after addition of the messenger RNA. Now it is before the addition of messenger RNA, transfer RNA binding is taking place. Transfer RNA binding is taking place. So this we call it 43S. This is, we call it 43S pre-initiation, okay, complex. Pre-initiation, so complex. This is the 43S pre-initiation complex. We can say PIC. Then, next we are coming to the second step. So, that is, we say second step. This is the second step. So, what happens second step? Preparation of messenger RNA. It is preparation of messenger RNA is taking place. So that is the second step. Okay. So in this, again, let us draw the messenger RNA. messenger RNA. We talk about the messenger RNA. The messenger RNA of the eukaryotic system. This is the messenger RNA of the eukaryotic system. Here it is AUG. It is 3' and here it is. So at the 5' there is a cap is present. Here it is 5'. Here 5'. Here it is cap. So 7-methyl guanosine triphosphate cap is present. So this is the messenger RNA. The preparation of the messenger RNA is taking place. In the preparation of the messenger RNA, a number of factors are added. What are those factors? The first factor is the factor which recognizes the cap. So that is what we call 4E. It is 4E cap. The binding of this cap is taking place. So binding of this cap is taking place. Okay, again, so let me write the messenger RNA. Okay, so this is the one. This is the messenger RNA. Here it is initiator codon. Here it is 3'. Here it is cap. Okay, it is 5' cap. Then the binding of a protein which recognizes this cap. So that is we say 4E. 4E bounded, it recognizes it. In the next step, here the binding of some other factors which are taking place. What are those factors? The factors which are bound to it are, let us see these factors. The factors which bound to are, okay, so the 4G, this factor is 4G, and then there is the binding of another factor. The another factor, it is 4A. The 4A, binding of another factor, that is 4A. Okay, here let us see what is happening here. Here, so binding of EAF 4G. So EAF 4G binds where? See, it binds EAF 4E and messenger RNA. That is the one type of binding. The second aspect is, and another factors they bind, that is EAF 4A. EAF 4A binds, okay, so EAF 4G and messenger RNA. Okay. This is messenger RNA. Okay, 3'. Here it is AUG. Okay, then this is the cap. So then the binding of cap recognizing protein 4E binding has taken place. Then there is a binding of 4G. The binding of 4G is taking place. The 4G binding, see here how the binding of 4G is taking place. Here one side it is binding 4E, another side it is binding the messenger RNA. So this is 4G. Then so there is binding of this particular, so 4A is taking place. Here it is 4A is taking place. This is 4A. Okay, so there are some important points. They shall be mentioned here. What are those important points? EAF 4G and EAF 4E. Okay, so binding, that is very important. These they control entire translation. If the initiation is controlled, entire translation is controlled. The EAF4E, how it takes place? EAF4E proteins competes with EAF4G. That is a way this control of the translation is taking place. In the next step, the binding of another factor is taking place. What is the factor? Another factor which is taking place. So what is the factor? The factor is, you can say 4B. The factor is 4B. Okay, so let us see the 4B binding is taking place. Along with this 4B, so how this messenger RNA complex looks like? How this messenger RNA complex looks like? Here it is. Okay, then here the binding, this messenger RNA, here it is AUG, and 3 prime, here it is the cap, okay, 5 prime, 5 prime, okay. Then there is the, that is the cap, then here it is E4E, 4E is binding has taken place, then, okay, okay, this is 4G, 4G, then, okay, then here 4A, 4A, then next is, okay, then here it is 4B, 4B, 4B. It is taken place. So let me explain at this point what is the significance of binding of this 4B. Significance of binding of 4B. The 4B stimulates, 4B stimulates helicase activity. Helicase activity. Which factor contains helicase activity? That is helicase activity of EIF4A. Once the factor activates helicase activity, the helicase unwinds. The helicase unwinds any secondary structure. Any secondary structure present in the messenger RNA. You can say secondary structure of messenger RNA. Secondary structure of the messenger RNA. Helicase unwinds any secondary structure of messenger RNA. So why the secondary structure has to be removed? So that association with 43S. So that association with, okay, 43, yes, PSC takes place. PSC, so association with 43 as PSC takes place. So that is the purpose of this, the binding of 4B activating it. So because of this one, what are the changes which are taking place? What are the changes which are taking place? Again, let me draw the messenger RNA along with the various factors. So this is the messenger RNA. So changes have taken place. Okay, then AUG. Okay, then this is the cap. Then here it is the 4E, that is 4E. Then there is presence of, see, because of the secondary structure, see how the changes which are taken place, 4G. Then there is the presence of A, 4A, then, so there is the B, 4B. These are the changes which have taken place. So, okay, let me draw again. So, here what happens? The 43S comes and joins. 43S PAC. 43S PAC comes and joins. Okay, 43S PAC comes and joins. Okay, let me draw this again. So, complex by joining, there is a formation of 48S pre-initiation complex. So, this is like this. Okay, then, so this is 3, factor 3, then, okay, this is 1A, then, okay, then here we have this one, okay. So then we have this particular one, this is 1 and this is 5, okay, then 5, here it is, methionine, then 2, factor 2 and GTP. Okay, then ultimately we have this. Okay, this is 3 prime. Here it is somewhere AUG. This is the cap. Okay, this is cap. Okay, then here there is presence of, here the cap is present here. Cap is present for A. Then there is the presence of 4G. Then so there is 4A. Okay, then here it is 4B. So this entire molecule, entire this diagram, this structure, what we say, complex. So we call it 48 as PIC. It is called 48 as PIC, pre-initiation complex. See, before going into this one, the association between the 40S complex, 40S ribosome and messenger RNA. So there is some rules are there. When you have studied in the prokaryotic system, so the proper association takes place at particular sequence. That sequence we call it Shine-Dalgarno sequence. Is there any such sequence in the eukaryotes? No. There is no sequence like Shine-Dalgarno sequence. But there is another type of sequence is there. So that is Kozak sequence. That is called Kozak sequence. Another type of sequence, so that we call it the Kozak sequence. It is a marker sequence. It is a marker sequence. Okay, marker sequence for identification. Marker sequence for identification. Okay, so that is the Kozak sequence, not Shine-Dalgarno sequence. Okay, then here when you talk about this particular aspect, let us go to the third step. What is happening in the third step? In the third step, the scanning of messenger RNA, by 40S. Why scanning is taking place? To find out the initiator codon. What is that initiator codon? The initiator codon is AUG. Further, what we require? Energy is required. So, okay, so there is energy in the form of ATP. ATP is converted to ADP. And what happens? The 40S subunit, okay, unit, along plus factors move along messenger RNA. So move along, okay, messenger RNA. So to find out that initiator factor, okay, that is AUG. This process is stimulated. This process is stimulated by EIF4, okay, A and B associated. What is that enzyme? Associated RNA helicase. That is RNA helicase. Okay, that because with the help of that enzyme it is taking place. So how the AUG, how they find it out? This is the AUG and this is the aspect, because the recognition of codon and anticodon. Anticodon in the transfer RNA. Okay, anticodon in the transfer RNA, codon in the messenger RNA. So let us see, after recognizing it, after finding the AUG codon, how this complex looks like. So again, this is the 40S. Then what I do is, first I will write this particular, okay, this is messenger RNA 3 prime, here it is cap, okay, this is the cap, here it is 5 prime, here it is AUG codon, okay, this is the AUG codon. Then I will be going on writing various factors. So first I will write, that is the transfer RNA, methionine, that is VAC. Okay, then there is the presence of 2 and GTP, then so there is, as it is, we have, this is factor 3. Okay, then so there is the 1A, here it is 1A, so then there is a 1, here factor 1, then here we have this one is, okay, then 5, it is 5. Okay, like that, which we have. Then next is, so there is, here it is E, that is factor E. Okay, then there is 4G, so this is 4G. Then next is, okay, then 4A, the next factor is 4B, like that. 4B. So the binding has taken place. So here it is, you can see 40S. Then ultimately the final 48S PSC has taken place with the proper alignment. Okay. In the next scenario, next is, it now goes to the step 4. So okay, then we can say it is 4th step. Here we can say it is 4th, okay, step. What happens in the 4th step? The 4th step is formation of ATS complex. Formation of ATS complex. Okay. So what happens? So once this correct base pairing has taken place, then only the association can take place. So that means we can presume the correct base pairing has taken place. Okay. Then what it will lead? It will lead to the conformational change. It leads to the conformational change. Okay. So in the conformational changes, where? In 48 PIC. Once the conformational changes which takes place in 48 PIC, then it will bring 2 changes. What are those changes? The first one is release of EIF1. So there is release of EIF1 is taking place. Okay. So then change in conformation of EIF5. Once the change in conformation of EIF5 and release of EIF1 is taking place, then this process stimulating what it does by stimulating. So EIF2 GTP is converted to EIF2 GDP. So once EIF2 GTP is converted to EIF2 GDP, so then EIF2, it is GDP, loss of EIF2 GDP is taking place. And simultaneously it stimulates, this process stimulates another protein factor, EIF5B. It also contains GTP. So this EIF5B GTP, it acts like, what is the function of this one? This one acts like prokaryotic, okay, IF2. What it does, prokaryotic IF2? It causes the recruitment of, see, in the prokaryotes, it is the 50S, okay, here it is 60S. Recruitment of 60S. In the prokaryotes, it is 50S. Okay, here it is 60S recruitment is done by the EIF5B. It acts like prokaryotic IF2. Before the recruitment of these 60S, there are some of the factors they are removed. What are those factors? The factors which are to be removed are factor 3. Also, So there is a removal of another factor. What is that factor? The factor is 1A. That is the 1A factor. So that is released. Okay. Then the next factor which is being released is the 5B factor. The factor which is being released is 5B factor. 5 factor. It is not 5B. It is EAF 5. That is EAF 5. Okay. Then, so there is an attachment of 5B GTP. Attachment of 5B, so GTP. 5B GTP is attached. EAF 5 is released. 3 is released. 1A is released. Okay. Next, in the next step, again, so the 5B is attached, and as I said, so there is attachment of, so the 60S attachment has taken place. Okay, then let us see here. Once 60S attachment takes place, then what happens here? 60S attachment so takes place. What happens here? So let us see how the ATS complex looks like. So let me draw the ATS complex. The ATS complex. Okay. So here it is the messenger RNA with various factors. So it will be attached here. This is messenger RNA with the, so cap, this is 5 prime and this is 3 prime. Okay, here somewhere we have this AUG is here. Then, so binding of this cap is taking place here. It is, this one is, sorry. So this one is, here it is taking place, the cap here attached here, E. The E is attached here. Okay, then, this is, you can say, so here E is attached here. Let us see, then E is attached here. Okay, this is E. Then we have this one. What we have here? Here, this one. This is the one. Okay, so this is here. It is, you can say, 4G. Then here it is. This is 4A. Then, so there is attachment of 4B. Okay. Then, so there is also, so there is an attachment of, so somewhere here, 5B and GTP. 5B and GTP is attached here. Then we have this A site. This is, what you say is, this is A site. Okay. This is A site and this is, sorry, this is B site and then this is A site. Okay, this is A site. Okay, so then here this is exit site, this is P site, this is A site. So at the P site, here it is, there is a presence of the transfer RNA, UAC, then here it is methionine, Then this one is 40S, and this is 60S. Next, there is another step, the final step which is taking place. What is it? Here, the 5B GTP is converted to 5B GDP. 5B GTP is converted to the 5B GDP. So, it is released. The 5B, okay, GDP is released. 5B GDP is released. Also, there is a release of E. There is a release of E is released, then also release of B. This is, B is also released. Also, so there is a release of, okay, so release of 4A. The release of the 4A is taking place. The 4A is released. Okay, then 4A is released. 4A is released. Okay, then also the release of the other factors. Other factors like 4G. Then final, we get the ATS complex. So let me draw the ATS complex. This ATS complex, here it is. Okay, then, so there is, okay, then, like this, 3 prime, this is cap, 5 prime, this is 5 prime. Okay. This is AUG, Okay, AUG. Then, so there is P site. This is P site, E site, then A site. P site, so A site and E site. In the P site, so there is presence of then UAC and methionine transfer RNA. Okay, then here it is E site, this is P site, this is A site, it is empty. This is empty. Okay, then this is 40S, this is 60S. It is 40S and 60S. Okay, then this is now ATS complex. ATS complex. ATS complex. Now it is for the elongation. It is for the elongation. Okay, let me summarize so far what we have learned in the eukaryotic initiation. Okay, in the process of translation. Okay, so the objective we have set is there are four steps are there that we have learned one by one. Okay, so before going to the detailed discussion, so as you know the translation, so there is initiation, elongation, okay, and so there is a termination process and also post-translational modifications. Okay, so what are those four steps? So binding of initiated transferrin to 40S, preparation of messenger RNA, scanning of messenger RNA by 40S, and formation of ATS complex. Then, when fresh round starts, so what happens? First is two events which take place, A and B. So in the A, ribosomal dissociation into 40S and 60S and release of messenger RNA. Initially 40S participates and converting to 43S initiation complex PIC, and then messenger RNA, many factors are added and binding with the 43 and 48 as PIC. Okay, then let us see what happens. So never the initiation starts with the ATS. It starts with only 40S. Okay, so in the presence of factor 3 and 1A, so the 40S they bind it, and this is the binding has taken place. The binding has taken place. Okay, with 3 and 1A. So first step, what happens? Binding initiated transfer RNA to 40S complex, then binding of factor 5 and 1 is taking place. Here I have shown it. Okay, in the next step, the 1A binding, so that prevents the binding of transfer RNA to A site because the post transfer RNA has to bind at the P site. Okay, after that every transfer RNA has to bind the A site. So the post transfer RNA shall not bind it. That is the reason 1A prevents it. Then, so there is a ternary complex, the combination of this transfer RNA, methionine transfer RNA, methionine, and EAF2 and GTP. Okay, so that is there and that binds it. Then this becomes 43S pre-initiation complex. In the second step, that is preparation of the messenger RNA. The messenger RNA in the eukaryotic system, there is a cap and tail are present. Cap 7-methylguanosine triphosphate cap. Cap is recognized by cap recognizing factor, that is 4E, which binds it. Okay, then binding of 4E is taking place, followed by the binding of 4G and 4E. EF4G binds EF4E and messenger RNA. 4A binds EAF4G and messenger RNA. This I have shown here. This also and this also. Okay. Then the entire initiation is controlled in turn translation by the EAF4G and EAF4E because the 4E proteins bind for the EAF4G site. Okay. In that way, it is controlled. Then later, binding of 4B takes place. 4B is the activator of 4A, 4A containing the helicase. Okay, so the helicase unwinds any secondary structure present in the messenger RNA that is necessary so that the association with the 43S PAC takes place. Okay, in the next step, so this 43S PAC and then messenger RNA with the various factors binding takes place, leads to formation of 48S complex, where you can see the many factors 1, 5, 2 GTP, methionine transfer RNA, 1A, 3, and 4B, 4A, 4G, and 4E. This is 48S PIC. So the correct alignment takes place due to the presence of Kozak sequence. There is a marker sequence for identification, whereas in prokaryotes, it is Shine-Dalgarno sequence. Then, in the third step, Scanning of messenger RNA by 40S is taking place to find out the initiator codon, that is AUG. It is an energy requiring process. ATP is converted to ADP and 40S subunit along with the factors moves along. So messenger RNA. So that is stimulated by EAF4A B associated helicase activity. So the association takes place because there is recognition of codon and anticodon, that is bases. Okay, then so this is the what it is happen. So the transfer RNA binds at the AUG1. Okay, then the next one is the fourth step, the formation of ATS complex. Okay, the correct base pairing. So conformational changes. The correct base pairing of codon and anticodon leads to conformational changes in 48S PIC. Okay, so then release of eukaryotic initiation factor 1, change in the conformation of the 5 that stimulates EAF2 GTP2, EAF GDP, that is release, and then that in turn stimulates EAF5B GTP. It acts like IF2 in prokaryotic system. So IF2 in prokaryotic system, it facilitates the binding of 60S and 40S. It recruits the prokaryotes, it is 50S and 30S. Whereas EAF5B, it facilitates the binding of 60S and 40S. So before binding, the factor 3 comes out, 1A comes out, this 5 comes out, and 5B GTP, it binds it. Okay, then so 60S also binding taking place. In the next one, what happens here? So after this binding, I have shown it. So various factors I have shown, and 5B. So 5B GTP is converted to 5B. So GDP, that is released, and E also released, B also released, 4A released. Other factors like 4G also, they are released. Ultimately, we get ATS complex. So this complex is having the E site, P site, and A site. So P site contains methanyl transfer RNA at the AUG initiator codon. A site is empty. A site is empty. Also, of course, E site is also empty. So this is 40S and 60S. Now it is ready for the elongation. It will go for the elongation. In the next session, we will discuss about the various aspects of elongation that are almost similar to the prokaryotic system. Okay, thank you. Thank you very much.